Remember my last video when I explained about glycolysis? I made the acronym PLOA. P stands for phosphorylation, lysis, oxidation, and ATP synthesis. So glycolysis is like the first step of cell respiration, and in cell respiration we know that there's something called the Krebs cycle. So we need to know what reaction links glycolysis and the Krebs cycle together. As the name suggests, it's actually called the link reaction. First of all, we have to know that glycolysis, it occurs in the cytoplasm. Well, where does the Krebs cycle occur? It occurs in the mitochondrial matrix. Where does the link reaction occur? It also occurs in the mitochondrial matrix. So we have to know, how does that pyruvate get from the cytoplasm into the mitochondrial matrix? Well, it gets transported from the cytoplasm into the matrix via a transporter protein, which is in the outer membrane of the mitochondrion. The summary of the link reaction is that it turns pyruvate into acetyl coenzyme A. How does that occur? It occurs through three processes. I like to remember it as DOB, kind of like Dobby from Harry Potter. Anyways, D stands for decarboxylation, and then O stands for oxidation, and B stands for binding. So in decarboxylation, as its name suggests, 3-carbon molecule pyruvate gets converted into a 2C compound. And then the second is oxidation. In oxidation, 2 electrons are removed and they are accepted by something called an electron carrier. In this case, the electron carrier is NAD+, and NAD+ turns into reduced NAD because it's accepting electrons. Remember oil rig? Oxidation is loss, reduction is gain. It gets reduced because it's gaining electrons since it turns from NAD to NADH. And then the third process is the binding of an acetyl group to a complex carrier molecule called coenzyme A. Hence, it produces acetyl coenzyme A. Now we can start the Krebs cycle. So, in this Krebs cycle, I want to start off with oxaloacetate. So oxaloacetate is a 4-carbon molecule, and then oxaloacetate gets turned into citrate. But how? It goes through a condensation reaction with acetyl-CoA via the enzyme citrate synthase. For IB students, every enzyme that I'm about to name in this video does not apply to your curriculum at all. Okay, so let's start. Oxaloacetate, 4C molecule, gets turned into citrate via a condensation reaction with acetyl-CoA via the enzyme citrate synthase. So in that reaction, that citrate synthase catalyzes, an intermediate is formed, which then releases CoA to yield citrate. And then I like to remember like the whole Krebs cycle as 665 4 times 5. 665 4 4 4 4 4. Number 1, citrate. Citrate gets turned into isocitrate. They sound similar, don't they? Well, that is because they are very, very similar. What just happened is that citrate goes through dehydration, and then it goes through hydration. What happens is that there's this hydroxyl group in citrate, and then the orientation just changes. That citrate to isocitrate conversion is catalyzed by the enzyme called aconitase. Now we're going to turn isocitrate into alpha-ketoglutarate. Now we're going from a 6 to a 5. So that means that there is a decarboxylation reaction involved. For every decarboxylation reaction in this Krebs cycle, you have to know that there is an oxidation reaction. So I just like to remember it as OD, oxidative decarboxylation. So in that case, to turn from isocitrate into alpha-ketoglutarate, NADH is produced, and so is carbon dioxide. And of course, the enzyme to turn from isocitrate to alpha-ketoglutarate is isocitrate dehydrogenase. Anyway, so now we're going from a 5 to a 4. So we are turning alpha-ketoglutarate into succinyl-CoA. Again, it has a decarboxylation reaction. Hence, it is oxidative decarboxylation. Hence, it would produce NADH and carbon dioxide. Of course, that is via the succinyl-CoA synthase enzyme. Then now we are going to turn succinyl-CoA into succinate. Now this is the part where ATP is produced. So of course, a phosphate ion is added to ADP, produce ATP, plus a CoA. Now we're going to turn succinate into fumarate. Now in that's just a redox reaction. So basically FAD is turned into FADH2 via the succinate dehydrogenase enzyme. And then we're going to be turning fumarate into malate. That's just a hydration reaction, so we're just adding water. It is catalyzed by the fumarase enzyme. And then we're going to be turning malate, finally, back into oxaloacetate. And for that is a redox reaction. So we're going to be turning NAD into NADH via the malate dehydrogenase enzyme, which catalyzes the reaction to turn malate into oxaloacetate. And that's it. Overall, for IB students, what you have to know is that in the Krebs cycle, one acetyl group is consumed. 3 NADs are turned into NADH, 1 FAD is turned into FADH2, then 2 carbon dioxide molecules are produced, 1 ATP is formed from ADP. So yeah, just be prepared, and if you have any questions, please comment down below. Thank you!